affects about an estimate about 2% of the population at around age 80, and about 850,000 people in the US. The average age of onset of Parkinson's is around age 60, but there is a lot of variation in the age of onset. So there are a number of people who will get it much younger. And so, I mean, you probably have heard Michael J. Fox, who developed Parkinson's in his late 30s. And so there is a lot of heterogeneity in the age of onset of Parkinson's. And so the main symptoms, motor symptoms of Parkinson's are tremors, typically a resting tremor where the arm is supported or it's laying at rest, rather than a tremor with movement. Stiffness or rigidity, poor posture, it falls, as well as slowness of movement. And really the slowness of movement and the postural instability later on in the disorder are the most disabling symptoms of Parkinson's. And so, before I move on to this slide, those are the main motor symptoms of Parkinson's. But Parkinson's really is a lot more than just a motor disorder. So for example, it affects speech, it affects swallowing. Many people have really bad constipation, sleep disorders, specifically what's called like a REM sleep disorder, where one acts out dreams, punches, thrashes, falls out of bed. Loss of smell, pain is a very prominent problem, sensory problems, urinary problems as well. So I don't want one to believe that Parkinson's is mainly a motor disorder. Really, actually, the motor symptoms for many people are the less bothersome symptoms. And when people have had Parkinson's for a longer period of time, it's really those non-motor symptoms, also including dementia, hallucinations, and psychiatric issues that really become the main problems of the disease. But this is a description of the motor presentation of Parkinson's disease. This was the original description, I think it was in the late 1800s by James Parkinson, who has the disease named after him. And he published what is called Essay on the Shaking Palsy, and that's why another name for Parkinson's disease is paralysis agitans, which means shaking palsy. And so his quote, actually I can't read it that well from this angle, but involuntary tremulous motion with lessened muscular power, with the limbs not in action, even when supported, with a propensity to bend the trunk forward and to pass from a walking to a running pace. The senses and intellect being unimpaired. I think that even today, having learned a lot more about Parkinson's than James Parkinson's knew, that's a pretty good description of Parkinson's motor symptoms. And this basically was just done from observation of 6 individuals, mainly one person, but also he had some brief interaction with 5 other individuals as well. So, so Parkinson's affects many people who you guys are probably familiar with. And so Linda Ronstadt, lead singer of the 1960s band The Stone Ponies. Andy Grove, former CEO of Intel. Brian Grant, NBA player. Michael J. Fox. Just one brief note. To the left of Michael J. Fox is Bill Langston, who is the founder of the Parkinson's Institute, where I'm working at right now. Muhammad Ali. Parkinson's doesn't have a clear cause. We don't know exactly what causes it, and so it's termed idiopathic. Means we're idiots because we don't know the pathology behind the disorder. So we do know some things that increase the risk, though, of Parkinson's, and so a lot of you probably are familiar with genetics as being a predisposing factor for Parkinson's, and so that's correct. And there are a number of genes now that have been identified as increasing one's risk of Parkinson's. It's not generally as simple as, okay, you've got the gene and now you're going to get Parkinson's. It's rarely that simple. So the genes increase your risk, your lifetime risk, of developing Parkinson's. There isn't full penetrance of the gene in causing Parkinson's, so if you have the gene, it's not a 100% chance. Some estimates are around 30% for example, a GBA gene mutation, and less for others. So clearly, genetics isn't the only factor, right, that comes into play about whether one's going to develop Parkinson's. The environment probably plays a role as well. And we'll go over some strong, some environmental factors that have strong evidence towards increasing the risk. But really the knowledge for environmental protective factors, the evidence for factors in the environment that protect against developing Parkinson's is pretty poor. There's some weak evidence that maybe coffee and tea may be protective, but really of all the studies that I'm aware of that have looked at either pharmacologic or non-pharmacologic treatment to prevent or decrease one's risk of developing Parkinson's, there hasn't been anything that has been proven to be successful. And so that's why there's a lot of interest right now in some of these disease modification strategies that we'll talk about later. But anyway, I rambled on, I'm sorry. So in terms of genetics, there are three main genes that can predispose to Parkinson's. One, the most common actually, is GBA, and so that's listed at the very bottom. When someone has two mutations in GBA, it causes a rare disorder called Gaucher's disease. It's mainly seen in the Ashkenazi Jewish population, and it's like a progressive childhood onset, severe developmental delay. But when one has a single mutation in this gene, it can increase the risk of Parkinson's. And now it's thought that this is the gene that is probably most commonly predisposing to Parkinson's. The other ones are LRRK2. This causes something that can be indistinguishable from idiopathic Parkinson's. It looks just like it. Starts around the same age as well, 50, 60 years old, and is fairly common as well. And then Parkin. Parkin is known for being the cause of juvenile Parkinson's. When two of the genes, both of the genes have Parkin are mutated, then this can cause childhood onset juvenile Um, Carolyn Tanner is a neurologist at UCSF, and she actually used to be the chief science officer at the Parkinson's Institute. She published some evidence that pesticides, manna, paraquat, increase the risk of developing Parkinson's. Really, that's the strongest evidence is for pesticides. And I don't think anything else really is convincingly associated in the environment with increased risk of developing Parkinson's. And so there's no proven restorative therapy for Parkinson's. There's only symptomatic drug therapy at the moment. And that's why really we would like to have something that delays the progression of Parkinson's. And so now, this is my slide that indicates that now it's going to get a little more technical. You know, I think that it's important to understand some of the science behind you know, these disease modification therapies. And if you guys are confused, you know, I still have trouble, you know, understanding a lot of the details behind all of this science, and it confuses me as well, so don't feel bad. But I think from here on out, no more fun of any kind, to quote Anna. Okay, so as we said, a disease-modifying therapy is what we want. This is the first step towards, I guess, a cure, you know, of Parkinson's. Something that can slow down the progression of the disease. Okay? And to date, everything has failed. Why is that? Well, we don't know exactly what the cause of Parkinson's is. I think a lot of studies have approached Parkinson's with the angle that there's one underlying disease mechanism. Well, that might not be the case. For example, a genetic type of Parkinson's can have a different mechanism than a non-genetic type of Parkinson's, right? And Parkinson's is not, as I alluded at earlier, is a very heterogeneous disorder, right? It starts at different ages for many people, and it has different speeds of progression for many people, and it has different variety, constellation of symptoms for many people. So there may not, for these different types of Parkinson's, there may also be different causes there. And so when people are trying to look at this, you know, maybe the population that they were studying, you know, was not the correct population. Maybe the population should have been just limited genetic causes or a specific subtype of Parkinson's. And so this is one challenge towards studying Parkinson's and therefore developing a disease-modifying therapy. Currently, most of the most of the interest in Parkinson's revolves around this protein called alpha-synuclein. So alpha-synuclein originally was a protein that was noted to be present in the brains of Parkinson's patients at autopsy, right? So a pathologist looked at the brains and he noted that there were these abnormal accumulations of these structures that were named Lewy bodies. And alpha-synuclein has been isolated as the main component of these Lewy bodies. And the thinking is that the alpha-synuclein is responsible for the destruction of brain neurons, brain cells in Parkinson's. The other alternative would be that it's a byproduct of the degradative process, right, in Parkinson's. But there is evidence that it does cause dysfunction of the cell. And so what I mean by that is that Parkinson's is characterized by a loss of dopamine-producing neurons, dopamine-producing brain cells, right? And the alpha-synuclein probably, through a number of possible mechanisms, causing stress to the cellular machinery, the components of the cell that produce energy, that use oxygen to produce energy, causes stress and dysfunction, of those structures. It can cause inflammation, an inflammatory response from the body's own immune system that therefore causes damage, to the brain cells. Problems with protein breakdown. There are structures in the cell as well that degrade proteins that you don't want to have there, and alpha-synuclein may impair that or interfere with that. And so it is not. So a lot of the strategies now are directed at removing alpha-synuclein or preventing the effects, the negative effects of alpha-synuclein. Two really popular studies right now that have a lot of attention from the movement disorders community are testing antibodies that are directed to different parts of the alpha-synuclein molecule. And so they bind to different parts, but the effect of the antibody is the same. It binds it, removes it from, hopefully from the brain, and therefore prevents the negative effects of alpha-synuclein. And so the two studies that are ongoing right now are Pasadena. This is testing an antibody developed by a European company called Roche, and then Spark, which is the American Biogen company. These are both, I think, available currently, or might be open for enrollment at UCSF and Stanford. I believe Spark is still open for enrollment. I think Pasadena might be closed, but I'll have to double check that. The alpha-synuclein antibody has been demonstrated to be safe, in phase 1 safety studies, and now they're testing it in a larger group of patients, hopefully for efficacy. One possible issue with this is that, it's not clear that it actually can get into the brain, right, and remove a significant amount of synuclein from the brain. And the brain really is where the motor impairment originates in Parkinson's. And so that may be one challenge, with this therapy. It's an infusion, it's a blood treatment, it's an IV treatment that has unclear effect on synuclein in the brain. There's no way to measure the alpha-synuclein in the brain as well. So that's another challenge, and we'll go over that. Currently there's not methods to measure the effect of disease-modifying therapy. Another possible way of targeting synuclein is blocking the clumping, the aggregation of the molecule. So as this diagram demonstrates, the synuclein really is dangerous when it clumps together, when it forms these oligomers. And so the goal for a drug called nilotinib, which is a leukemia drug that's used in CML, a form of leukemia, is to block the clumping of synuclein and therefore hopefully delay the progression of Parkinson's. I know this was being studied at Oregon Health and Sciences when I was working there about a year ago. So other ways of targeting synuclein include increasing the activity of this enzyme called GCase. Now, GCase is an enzyme that breaks down bad protein. They're called glucosylceramides. And really, this enzyme is abnormal in people with a GBA mutation. So I mentioned earlier that GBA is one of the genes that predisposes to Parkinson's. And this gene is not as active in those people. And actually, even if someone doesn't have a GBA mutation, it has been shown that people with Parkinson's, idiopathic Parkinson's, may have lower GCase activity. And so this enzyme may play a role just in regular Parkinson's as well. And so if this enzyme is not working well enough, then it may increase the synuclein in the brain and therefore worsen, cause Parkinson's. So one strategy is to increase the activity of this, and there's a drug called ambroxol that is being studied that does this. Another way is to reduce the production of the protein that GCase breaks down, right? And so if there's less of that, there's going to be less buildup of possibly this toxic protein, and that may be protective. And so we're studying actually at the Parkinson's Institute this drug called venglustat. This is a drug that reduces the production of these glucosylceramides. And so right now, we're enrolling people with GBA mutations, demonstrated GBA mutations, and providing this drug to see if it can delay the progression of motor symptoms. And then also another targeted therapy. We didn't spend too much time on LRRK2, but this is another common drug that's mutated in people with genetic causes of Parkinson's. And so there's an LRRK2 kinase inhibitor. Basically, all you really need to know about that is that LRRK2 kinase, blocking that enzyme is thought to be a potential mechanism for slowing disease progression in Parkinson's. So there's drugs. Denali Therapeutics is a company that's testing drugs that do that. Have I lost everyone yet?
Yes.
So I told you there's going to be no more fun, right? So another reason that we've had trouble developing protective drugs for Parkinson's is that animal models may not represent well human Parkinson's, right? And for example, a mouse model where they do a mutation to the synuclein gene to cause Parkinson's doesn't have the same pattern of synuclein as a human Parkinson's patient does. And so that's another reason why the research has had trouble in developing a cure or a disease-modifying drug for Parkinson's. Another main reason is that there's no way to measure the effect in the brain of disease-modifying treatment. You can't measure the synuclein in the brain, and that's a huge challenge. So how do you measure the effect of a potential disease-modifying drug? Right now, what's really being done is just having a clinician perform an examination of a patient and documenting the motor symptoms, right? Well, is it possible that the drug being studied is affecting some of the dysfunctional mechanisms in the brain, but not quickly enough for that to cause a symptomatic improvement, right, in the motor exam? Yes, that's possible. It may take time for any specific disease-modifying drug to result in a change in one's motor function, even though the underlying, dysfunctional mechanisms may be corrected. So that's one issue. There is that a clinical exam is usually the outcome measure for most of these research studies. There also is a scan that one can do to look at the amount of basically the dopamine-producing neurons in the brain. Right? There's a scan that one can look at. It's called a DAT scan. It measures the activity of these dopamine-producing cells. But the technology really needs to be improved for research purposes. Often what's done is, someone looks at signal intensity on the computer screen and compares them. If one side, if it's lower, then it's thought to be abnormal. So it's a subjective analysis. Has anyone heard about PPMI? PPMI is a biomarker study that's being done by the Michael J. Fox Foundation, and I think it's pretty important. So, as I said, we need reliable biomarkers that can measure the progression of Parkinson's, the impact of treatment on Parkinson's, and this is something that's aiming to do that. And so we're doing this at the PI. A lot of other places, academic institutions are hosting this as well. It's not an interventional study, so it's not testing a specific drug for anyone, but it's an observational study that is collecting information that will be useful in the future for hopefully developing a cure for PD. And so what they're doing is they're getting people with Parkinson's, with genetic causes of Parkinson's, and people who don't have Parkinson's. Maybe someone, or maybe are healthy controls who have risk factors for Parkinson's and who don't have risk factors. So, and they're collecting blood samples, they're collecting spinal fluid samples and doing examinations on these people to hopefully identify something, a measure, right, a biomarker that we can use for future research. So I think this is going to be pretty important for developing a cure for Parkinson's. Okay, so I kind of mentioned this other challenge, right, in developing disease-modifying therapy for PD. The outcome measures that we use are not very good. That's why we need reliable biomarkers. And so the neurologic exam is probably inadequate for being the outcome measure for disease progression. Another problem is that early Parkinson's already has it's thought that when someone develops motor symptoms of Parkinson's, it's thought that they've already lost about half of their dopamine-producing brain cells. And so it may be too late to provide disease-modifying therapy into someone after they develop the motor symptoms of Parkinson's. And so what may need to be done is you have to identify people in the premotor state. And so I mentioned some of these symptoms here that are non-motor symptoms of Parkinson's: loss of smell, constipation, sleep disorder, REM sleep disorder. Identifying these people before they develop motor symptoms of Parkinson's may allow us to provide potential disease-modifying therapy before there's been too much loss of dopamine-producing brain cells. And so maybe we just need to start treating premotor Parkinson's as well. And so I did want to mention that has anyone heard about the gut-brain connection in Parkinson's? Yeah. And so before the synuclein deposits in the brain, it may deposit in the GI system. And so that might be the reason that someone develops constipation a decade before any motor symptoms of Parkinson's manifest. And so it's thought that one hypothesis is that the synuclein maybe travels from the gut up one of the nerves to the brain, and then reaches the brain, and then one develops the motor symptoms of Parkinson's from there. And so, if you can identify someone with GI issues before they develop the motor symptoms, those people might be the ones that will respond best to a disease-modifying drug. And so we're doing a study at the Parkinson's Institute. It's called the GI Natural History Study, where we're looking. It's basically a biomarker study like PPMI is just for GI symptoms. And so we're looking at people with Parkinson's, without Parkinson's, but at risk for it, and then following them over a number of years, and then measuring through endoscopy and through a pill that's swallowed, measuring the function of the GI tract. And so hopefully we'll obtain information that will allow us to identify characteristics of the GI tract, right, that will point towards a premotor state of Parkinson's. Okay, so that's a lot of scientific information. I think that the bottom line that I want to get at is that there's a lot of research that is underway at trying to develop a cure for Parkinson's. And it's not going to be easy, right? I mean, if anyone says that, they're probably misleading you. And there's a lot of challenges towards developing a cure or a way to at least slow down the progression of the disease. We're working on it though, and I think that these strategies, these studies are hopeful, and that we will have some progress, in the next couple of years. Any questions? Yeah. Yes.
What about stem cells?
Get a microphone. Now everybody can hear. I will work my way around. Make sure you get your hands up.
What about stem cell research?
Yeah, so I've been in movement disorders and seen Parkinson's now for about 4 years, and it hasn't been a popular avenue since I've been in this field. I know it was really popular, like a decade or two ago, and there's a lot of news on that, but the interest has fallen off. There's maybe some renewing interest in these fetal stem cell grafts, but nothing very promising at the moment.
Right here.
Do you believe, with all of your experience and your part in what the families can do to keep the Parkinson's at bay with activities and exercise and cognition, activities, etc.? Do you believe that the Parkinson's can sort of be kept at bay if this is done regularly and diligently in the Parkinson's patients?
That's a good question. So the exercise is probably effective at reducing the progression of motor symptoms in Parkinson's. I'm not aware of any evidence that it'll slow down the loss of dopamine brain cells, but especially in early Parkinson's, doing intensive exercise, where one exercises to about 80% of their maximum heart rate. For example, there was one study that looked at treadmill running, and they compared people who did intensive treadmill running to 80% of their max heart rate, to people who did about 60, a moderate amount, and then 40%, or not very intensive. And this was all in people who were untreated, really early Parkinson's. Over a 6-month period, the people who did really intensive exercise, 80% of their max heart rate, on a treadmill 4 times a week for I think about 30 minutes a day, they didn't get worse in terms of a clinician measured examination. So, I do think that, I do always, recommend it for everyone because exercise is amazing, it's great. I'm just saying that I'm not sure if it slows the loss of dopamine producing brain cells or the underlying problem in the brain.
Right here?
Yes.
I've had DBS surgery, and it was incredible. I mean, probably 2 years ago, I would be sitting at this table going like this, and now I'm completely tremor-free, and it's a great success. But my question for you is, it seems like the questions that are being asked through the clinicals, you can ask what you know about the disease, but like freezing, it happens to me 2 or 3 times a week, and sometimes more. Is it possible that they should be looking at the individual disease as something separate? Maybe it's not Parkinson's, maybe it's Jim's disease. So can you comment on that in terms of treatment? Am I way off base, or is there a possibility to look at freezing, which is very common, and treat it like a separate disease, and go after it like a separate disease? It's different.
Okay, so is the question how do you treat freezing in Parkinson's?
That would be part of the answer, I think, but my question is, I do things that they don't seem Parkinson-like, and so I wonder if maybe they're barking up the wrong tree. I don't know.
Forgive me, I'm not sure I really understand the question, but there is a lot of variety in the symptoms of Parkinson's, and freezing is something that can occur in Parkinson's, right? But also in other related disorders, atypical Parkinsonian disorders, and so it's a manifestation of probably a problem both with the dopamine in the brain as well as the frontal lobes. The frontal lobes are involved with planning, with multitasking, and attention. And walking is a very complex movement, right? I mean, walking, if you think about it, requires input from your sensory system, it requires cognitive function, it requires the ability of the basal ganglia, the dopamine producing cells to initiate movement, strength of your muscle. So it's a very complex movement, and so there's lots of things that play into walking. One thing that's coming into attention is that one's cognition plays a huge role on freezing. Have you noticed that if you're focused on something else, or if someone talks to you, that you might freeze, right? And so currently, some of the strategies are using actually cognitive enhancing medication to treat freezing, not treat freezing, but to treat loss of postural stability. And so falls, for example, in Parkinson's, may be treated with the medication that has been used to treat dementia. And so I've been using that myself recently. The Movement Disorder Society has recommended that there's fair evidence, for these drugs in treating loss of postural stability in Parkinson's. Freezing is different, though. It doesn't typically respond to these cognitive enhancing drugs. Did that answer your question, or was there something else that you wanted to know?
Right here.
Is there any research being done using amino acids together with drug therapy?
Amino acids. Can you clarify that? What specifically?
The use of amino acids to slow the progression.
I haven't seen that. There may be something like that. I haven't seen that. Yes.
Hold on, guys. I have a line.
And there's only one of me.
Alright, we go here, and then, wait, so is it you, Sandra, first? Okay.
Okay, you had mentioned scanning the brain with the DAC scan or DAC. Would you spell that, please? Because you just kind of covered it and then moved on.
So there's a scan. It's spelled DAT, and what it is someone comes into the MRI scanner. They're injected with a chemical that then is taken up by the neurons in the brain that produce dopamine, right? And so when this chemical is taken up by the dopamine-producing brain cells, it lights up on a brain MRI. And so one can, with this scan, measure the amount of dopamine-producing brain cells. If it's lower, if it's not lighting up as brightly as it normally does, well, that indicates that you've had loss of dopamine cells and that you may have Parkinson's disease. And so it's a useful test if someone is questioning whether this is Parkinson's at all, or whether this is, for example, essential tremor or some other cause of tremor. It's a useful test to clarify whether someone has Parkinson's.
Right here.
Is there any long-term benefit to intravenous application of like glutathione or any other of the other so-called brain supplements that are available?
Yeah, I haven't seen any strong evidence on that. I know that's being done by a lot of these clinics out in the community. Glutathione is supposed to be an antioxidant, and so that's how it's proposed to work. But I haven't seen any strong evidence. The Movement Disorder Society hasn't recommended it as a disease-modifying treatment.
Right here.
Thank you for taking our questions. One is about red light therapy. I understand that there's something being done, I think it was in Harvard, where the red light is inserted maybe up the nostril to the base of the substantia nigra, and it increases the mitochondrial development, which helps in there. Do you know, have you heard of that? Know anything about red light therapy?
I have not heard about that.
Okay.
And now, then I've got somebody back here. I'm coming, guys, I promise.
You mentioned Lewy body in your talk. Would you talk a little more about that?
Yeah, sure.
Is that something that's before Parkinson's, after Parkinson's, that kind of thing?
Yeah, sure. So, a Lewy body is a structure, abnormal structure in the brain of people with Parkinson's when you look at the brain under a microscope. This is really how Parkinson's is diagnosed, actually, right? I mean, at autopsy, you have to see these structures. And what these structures are made up of, as I said briefly during my talk, was it's made up of alpha-synuclein. So what pathologists can do is they have a stain for synuclein. They stain the brain with it, and any synuclein will light up with it. And if there's a large collection of synuclein, that may be a Lewy body. Now, there also is another disease that's related to Parkinson's called Lewy body dementia. And the main difference between this and Parkinson's is where the Lewy bodies are located.